Hello guys, this video will going to be all about on alternative or Properdin pathway. This is the last pathway of complement system. Okay, so again this pathway is antibody independent. Okay, so as it is antibody independent pathway, so we can say it is also an effector arm of the innate immune defense. Okay, so now you may probably think that as I have here written alternative pathway or Properdin pathway, then I think you may think that Properdin will going to be the play the role of the activator, as we have seen in case of the lectin pathway. But here Properdin is not the activator. Okay, this alternative or Properdin pathway can be activated by almost any foreign substances. Okay, for example, let's say lipopolysaccharide from the outer membrane of the gram-negative bacteria, the cell wall of some yeast, a protein present in cobra venom known as cobra venom factor, along with that some activators that can also activate classical pathway here also play the role of activator. For example, viruses, aggregated immunoglobulin, necrotic cells. Okay, although here I have taken only some examples, but any kind of foreign substance can activate the alternative pathway. Okay, so classical pathway is an example of humoral adaptive immune response, and both lectin pathway and our alternative or Properdin pathway are the examples of innate immune response. Right? Please note this very carefully, and it is very important for your MCQ exams. Now, your lectin pathway is activated by lectin. Okay, any kind of bacteria or any kind of antigen that have terminal mannose residue in its surface can be destroyed by lectin pathway, and alternative pathway can be activated by any kind of foreign substances. Okay, so now here four serum protein will going to play important roles. One is C3, factor B, factor D, and Properdin. Here factor D is the cleaving protein. Factor D have enzymatic functions, and Properdin or factor P, on the basis of which this pathway is named as Properdin pathway, this Properdin will going to play the role of a stabilizer. Okay, so now I hope it up to that point the topic is clear, right? Now let's move to the steps. Okay, so now we will going to talk about the steps of the Properdin pathway or alternative pathway. So it all begins with the spontaneous hydrolysis of C3. Okay, here the hydrolysis of C3 is not facilitated by any kind of serine protease. Okay, it is a spontaneous hydrolysis, and as a result it will break into two fragments, C3a and C3b. Okay, and as we have discussed, C3a will be the smaller fragment and it will be dissociated. C3b will be the larger fragment and it will take part in the further reaction. Okay, now this C3b will bind with the foreign surface antigen in the step 2. Okay, that means after the spontaneous hydrolysis of C3, the C3b will bind with the foreign surface antigen. Now, after binding of C3b on foreign cells, C3b will again bind with another protein named factor B and leads to the formation of a complex. Okay, this complex includes C3b, factor B, and the foreign cell. Okay, now after binding of C3b, factor B becomes the substrate for factor D. Okay, that means C3b facilitates the cleavage of factor B by factor D in the next step. Okay, and factor D will going to cleave factor B into two fragments. One is B small b and B small a. Okay, again B small a is the smaller fragment, so it will be dissociated, and B small b is the larger fragment, so it will facilitate further reactions. Okay, so it all starts with the spontaneous hydrolysis of C3 into C3a and C3b. C3b will bind with the foreign surface antigens. Okay, upon binding, it will also bind with the factor B and leads to the formation of a complex containing factor B, C3b, and foreign surface antigen. Okay, and after that, due to the binding of C3b, the factor B becomes a substrate for factor D. Okay, and factor D will cleave factor B into B small b and B small a. Okay, and the B small b will going to facilitate further reaction, and B small a will dissociate. Okay, now this foreign surface antigen C3b and B small b together is the C3 convertase. Okay, and this C3 convertase will be stabilized by the properdin. Okay, so in alternative pathway, properdin plays the role of a stabilizer, and it stabilizes C3 convertase. Okay, so this is the end of this video, although the pathway is not ends here, because as I have told you in earlier videos, we will only focus on the formation of the C3 convertase, because the rest of the part is same for all the three pathways. So I will going to take one video to explain the rest of the part that is common for all the three pathway: classical pathway, your lectin pathway, and alternative pathway. Okay, they are differing from among each other only up to the C3 convertase formation. Okay, so I hope this video will be helpful for you. So thank you for listening to this class.